Now we are welcome back to the class on cell and its environment. This class will be describing a simple experiment on osmosis. And don't forget that osmosis is actually the movement of water molecule through a semi-permeable membrane from a region of higher water potential to a region of lower water potential. Now, in this case, so many experiments could be found on the net, anywhere, in textbooks and co. But just to describe experiment on osmosis in a simple way, the semi-permeable membrane could be any living tissue. Any living tissue could act as a semi-permeable membrane. And in the case of this particular experiment, I try to make use of peeled yam. Peeled yam, hollow shape created within the peeled yam. So this is yam tissue. That's what it has been peeled, then we make a hollow shape within it to serve like a cup to use for this experiment. Now in A, I put some water, some solution of salt. Salt solution was put inside hollow shape A, and that salt solution is ensured to be hypertonic in nature. Hypertonic. Why is in the second yam tissue no solution was placed inside of it? Then around the tubers of yam, or the tissues of yam, or the solution of was distilled water we are placed there. This distilled water acts as the hypotonic solution, while the salt solution acts as hypertonic solution. So we have hypo and we have hyper. This external environment of the yam tissue is acting as hypotonic, no solution, hypotonic solution, while the internal environment now, which is the hollow shape that has been created inside the yam tuber, is acting as the in it or salt solution acting as hypertonic solution. The setup was left for some period of time, just as shown above. After 12 hours, it was observed that the water level in the level of the solution in A rises, while the one in B, no water comes in, no solution comes into yam tuber B. The one in A, the rising of the solution in A is just describing that water solution, water, it's a water molecule, has succeeded passing through the yam tissue into the salt solution that is hypertonic. And this is describing simple osmosis in the environment of the cell. So the cell, which is a semi-permeable membrane, allowed water to percolate because the internal environment is actually hypertonic. So just to buttress the point that osmosis involves the movement of water molecule from a region of higher water potential. In this case, the distilled water is a solution with higher water potential, while the salt solution is a solution with lower water potential. So water has actually moved from the distilled water through the semi-permeable membrane into the salt solution to raise the level of the salt solution in A. So the osmosis occurs in different tissue, in so much that there is a margin or a difference in the concentration gradient of the two environments. Now the order of the two environments. Now we take time to explain the other events that occurs. The other events that occurs around cell or within cell, as the environment of the cell is not same as that of the internal environment of the cell. So whenever the external environment of the cell is not the same with that of the internal environment of the cell, other things that are expected to happen are plasmolysis, crenation, flaccidity, turgidity, and Haemolysis. So we'll quickly pick them one after the other to have a better understanding of what would happen to a cell whenever the internal environment differs from the external environment based on concentration. So whenever there is concentration gradient difference, there will be movement of molecules. Now in these parts from Haemolysis down to from plasmolysis rather, down to hemolysis. There are two terms I want us to understand very well, and these terms are endosmosis, and exosmosis. So the two words from the word osmosis, we should have a very good understanding of what these two words will be. Since we already know that osmosis is the movement of water, so endosmosis in this case now will be movement of water into the cell environment, while exosmosis is the movement of water out of the cell internal environment. So when water is moving into the cell internal environment, we say it is endosmosis. When water is moving out, water molecule is moving out of the cell environment, we say exosmosis is occurring. Now, plasmolysis, to explain it, plasmolysis is just an event that occurs in a cell when there is exosmosis. When there is exosmosis, the cell can become plasmolysed. That's the word. The cell can become plasmolysed. And plasmolysis occurs when there is shrinking of the cell vacuum to the extent that the cell membrane detaches itself from the cell wall as a result of shrinking of the cell vacuum and also the cell cytoplasm. And this will only occur when there is excessive loss of water from the cell internal environment. So exosmosis will result in plasmolysis. Now, crenation is also an event that occurs when there is exosmosis, and this will occur when there is excessive loss of water within the cell as a result of the cell being placed in an hypertonic external environment, hypertonic solution. The cell being placed in an hypertonic solution and resulting in the cell forming a kind of undulating end. The end of the cell started forming a kind of crenation. It is no longer round as it used to be, or forming a kind of regular shape. But in this case, the air became serrated, started forming a kind of serration or a kind of undulation. There's a serrated end because it has lost a lot of water. We call that crenation in biology. Then the next one is flaccidity. Flaccidity is also similar to plasmolysis, although not the same. It occurs also when there is excessive loss of water from the cell vacuum or the cell cytoplasm. But in this case, the cell membrane is not detached. It's not detached from the cell wall. Although there is excessive loss of water, there is exosmosis within the cell environment. The cell environment, the cell internal environment loses a lot of water in the case of flaccidity, and flaccidity can result to wilting of leaves, falling off of fruits, and so many other events in plants. Then the next one, which the last two are actually product, or they are events, they are phenomena in cell that occur as a result of endosmosis. That means movement of water into the cell environment. Movement of water into cell environment, into cell internal environment. When there is movement of water molecule into the cell internal environment, we say endosmosis has occurred. Now, turgidity. Turgidity on its own is actually the movement of water into cell, resulting in the cell vacuum being filled up with water, then pushing the cytoplasm and the cell membrane, and the cytoplasm and the cell membrane add on the cell wall. When there is much inflow of water into the cell internal environment, resulting in large vacuum pushing the cytoplasm and the cell membrane add on the cell wall. This turgidity can actually bring about what's called Turgor pressure. Turgor pressure. Turgor pressure. Turgor pressure is actually used by plants to make its leaf, flower to stand upright, to stand well during the day. So Turgor pressure is actually very, very important in plant. It will allow plants parts like the leaf to stand upright during the day. Turgor pressure is also very, very important in the cell of parenchyma cell. Parenchyma cell, they become a supporting tissue in plants whenever they are turgid. Whenever they are turgid, they become a supporting tissue in plants. Now the last one is hemolysis. What hemolysis? The word hemo has something to do with blood, while lysis, as I've said in the previous class in biology, means breaking down. So hemolysis is actually the process by which red blood cell swells and bursts as a result of excessive inflow of water. So when there is excessive endosmosis, the red blood cell will swell and burst. In that process, we say hemolysis had occurred. Hemolysis had occurred. So we have all of these that we have mentioned. They are actually passive transports. Why do we say passive? It is simply because the use of ATP is actually not needed. You don't need ATP. No ATP energy is not needed in most of this event. But when we come to active transport on its own is the active. Yes, active transport. Active transport. So active transport will always require energy in form of ATP because it is a movement of substance. It is a movement of substance, a molecule of substance against concentration gradient. You are moving against concentration gradient just like you are moving up and hill. You require more energy to do this than when you are moving down a slopey environment or hill. So active transport is actually movement of molecules against concentration gradients. Movement of molecule against concentration gradients. In the next class, we shall be looking at other things, and we shall be looking at some exercises and some life phenomena that actually describes most of these events. Thank you. An assignment will be given to you, which I will expect you to drop the answers on the comment section. And the assignment reads thus: when the spirogyra cell is immersed in a salt solution more concentrated than its cell sap, it A. remains unchanged, B. takes up water and bursts, C. absorbs little water, D. loses water and shrivels. Jam question 1983, question number 47. A similar question was also asked by WAEC in the year 2001 and 2015. Just similar question. The only thing we have there is the options we are differs where we have when the spirogyra filament is placed in a concentrated salt solution for 30 minutes, the cell will become, we have option plasmolysis, turgid, shorten, elongated. So it's almost similar question. So the second question we have on the board, which I'll be expecting the answer from the comment section, is turgidity in the cell of plants does not easily lead to bursting of the cell because then we have options. A. Cell membrane can resist the turgidity. Cell membrane can resist the turgidity. B. Large vacuole contained in the cell can continuously take in water. Large vacuole contained in the cell can continuously take in water. Then C. Cell wall can resist the turgidity. C. Cell wall can resist the turgidity. Why D. Cytoplasm is jelly-like. That's WAEC question 2001 question number 39. I invite you to like and share the video. Meet you in the next class.